Yes, my name is Brian Thacker. I'm a scientist in the CMC group at Bioanalytics. I have 10 years of experience in drug development industry, with many years at an early stage startup where I was doing cell line development and assay development, and then joined Bioanalytics a few years ago, again in the CMC team. Next slide. As we consider the CMC approaches and the control strategies that we might use for ADCs, I just want to take a minute to step back and review the composition and the synthesis of the ADCs, because that informs the parameters, the attributes that we need to monitor and in a control strategy. So as you know, the starting material for the ADC is a monoclonal antibody, which in and of itself contains quite a bit of heterogeneity and complexity, with multiple protein chains and post-translational modifications. To create the ADC, the antibody is chemically modified to introduce the linker molecule, and then followed by chemical modification to introduce the drug. And these chemical modification processes are well controlled, and the technology is improving all the time. But the chemistry itself gives rise to another layer of complexity and heterogeneity, which needs to be monitored and accounted for in a control strategy. So with that, that basic understanding, let's go to the next slide. And this is a complex table, but just not to go through all the details, just to point out the various different attributes that can be monitored or should be considered for monitoring in a control strategy for an ADC. Some of these attributes would be monitored for any large molecule drug that's in production. But then there are some structural features, particularly that are specific for an ADC, such as the drug-antibody ratio, the conjugation position of the drug molecules, presence of unconjugated antibody, and glycosylation, to name a few. We're not going to touch on all of these different parameters, but we are going to show some case studies in the next few slides for the ones that are highlighted here in yellow. And just before we go to the next slide, I just want to point out that that the complex structural features of the ADC require sophisticated technologies to capture all of that variance and complexity. And so what I'll be showing is analysis with high-resolution mass spectrometry to identify and monitor those structural characteristics of the ADC. So with that, let's go to the next slide. Just a brief note here on the ADC and how it can be analyzed using mass spectrometry. Certainly, we can analyze the intact molecule using mass spectrometry, but the complexity inherent in this molecule can make it difficult to interpret the data and to tease out the different structural features that we'd expect to find and we need to monitor. Fortunately, there's some fairly simple ways that we can break down the ADC and produce subunits that are more amenable to analysis by mass spectrometry. And I've got that highlighted on here just to illustrate those approaches. Digestion with the enzyme IDEs produces a very specific cleavage at the hinge region of the ADC. So that liberates the FAB2 portion of the antibody from the FC portions, again, breaking it down as a way to reduce the complexity of the ADC. We can also reduce the molecule using a reagent like DTT to eliminate the disulfide bonds, and that would further break down the subunits and separate them from each other so we can look at them independently and characterize and monitor the structural features of each of those. If we go to the next slide, we can look at a short overview of the of sample preparation methods and then analysis of ADCs. So as I mentioned at the top there, intact ADCs starting material. It can certainly be analyzed directly using LC-MS, or it can be digested using the IDES enzyme, which is a simple digest at 37 degrees, breaking it down into some component subunits, and then further reduction using DTT to complete the breakdown into subunits. Any of those preparations can be loaded onto a liquid chromatography system attached to a mass spectrometer. In this case, we're using a thermo Orbitrap Exploris and taking advantage of the high mass resolution features and capabilities of the mass spectrometer to collect high resolution mass data. The data that comes out is really complex. You get a complex mass spectra. It's difficult to interpret the mass spectra directly, but we can take advantage of software packages like Biopharma Finder that can be used to deconvolute the data, extract the masses that are present in each sample. And then by informing Biopharma Finder of the expected amino acid sequence of the antibody and the expected masses that could be found either in post-translational modifications or as a result of chemical modification, the Biopharma Finder can align the masses found in the sample with the masses that are expected to be found and annotate those masses for us and give us relative abundance of those masses. We can also use the process to look for spurious chemistries, incomplete conjugation reactions, other spurious chemistries that would be impurities that we want to monitor during the in the control strategy. Let's move to the next slide, and I'll show you some examples of the data that can be collected using this kind of approach. This first slide here is showing an example of intact mass analysis. So in this instance, the antibody hasn't been modified at all. It's been loaded directly to the LC system. In this instance, it's a reverse phase column. And we have the Orbitrap set to high mass range to accommodate the large mass of the ADC that's coming into the system. Some representative data is shown on the right, and I'll just walk through this figure to orient people who might not be familiar with this kind of analysis. On the x-axis, we have the mass that's being detected. That's mass in Daltons, so you can see we're working up in the 150 kilodalton range, which is what you'd expect for an intact antibody. The y-axis is relative intensity. So we're looking at the abundance of those masses within the sample. Each peak that you see along the x-axis there is a discrete mass that's been identified by the mass spectrometer. And then Biopharma Finder's been able to annotate those masses and identify the structural, where they come from structurally in terms of the material that's in the sample. Each red triangle is an annotated peak, and then some of them have labels on them. Too many peaks here to put all the labels on, but you can see maybe looking at that highest peak there, the D3 ADC, that's identified the ADC molecule in there. And then the annotation underneath that indicates the glycoforms, the glycans that are present with the G0F and the G1F glycans, and then the number of drug molecules. In that instance, it's 3 drug molecules per antibody. Okay, so I just want to point out 3 observations from this data set. There's lots and lots of data here. You can see that there's lots of masses that have been identified, lots of different structures that are present in this complex mixture, which is just the intact antibody. We look at the major peaks or the major signals in there, you'll see that there's triplet signals that kind of repeat in step order fashion across the masses on the x-axis. If we look at those triplets, we can see that there's different glycoforms represented in those triplets. So I like the oval there that has the dotted line around it. That's highlighting one of the triplets. And the top peak there, which is annotated, you can see has two glycans found on that ADC, both biantennary and glycans carrying the fucose, but they vary in the presence of galactose. So you see that there's one G0F glycan attached to that antibody and one G1F glycan attached to that antibody. That's the major peak there, but if you look to the peak just to the left, to the lower mass peak, that corresponds to 2 glycans that are devoid of the galactose, and then the peak to the right has both of those glycans carrying the galactose, showing up at a higher mass. So we see those triplets repeating across the mass spectra. Why do they repeat across the mass spectra? Well, we're seeing evidence of different conjugation levels of the ADC. And so on the far right, you see D equals 0 labeled underneath the x-axis. Those are instances of monoclonal antibody that have no drug conjugate attached to them. So it's bare, naked antibody that is found in the sample. That's on the far left. I might have misspoke. That's on the far left of the x-axis. On the far right, we see that drug ADC that has 8 drug molecules attached to the antibody. And then there's a distribution of drug attached to the antibody. And most frequently, we see 3 or 4 drug molecules attached to each antibody. Biopharma Finder can take all of this data and calculate an average drug antibody ratio, which you'll see at the top of the figure there is calculated in this instance is 3.07. So we have approximately, on average, 3 drug molecules attached to each antibody. Okay, let's go to the next slide. Here we're showing some representative data of an ADC after it's been digested with the IDES enzyme. Again, this is an enzyme that cleaves very specifically at the hinge region of the antibody and liberates the FAB2 portion of the antibody from the FC domains. As you can see from the figure on the right, the complexity of the masses that we see is quite a bit reduced. If you look at the annotation on the major masses, the big peaks that are shown there, you can see that we see evidence of the Fab domain with no drug linked to it, all the way up to 7 drug molecules on the far right, the x-axis. Again, with the most abundant conjugation status for the FAB being the 2 drug molecules. An approach like this simplifies the analysis, and it also gives us opportunity to see other features that might have been obscured in the intact analysis of the ADC. And in this point, in this data, I'm just pointing out that we can observe and monitor some of the product impurities in the ADC. So what I've boxed down below with the dashed line rectangle are some lower abundance masses that are detected in the sample. These masses correspond to ADC that has linker attached to the ADC, but is missing the payload. So for instance, that peak on the far left, that's the lower peak in the box, that's an ADC that has 1 complete linker and drug molecule attached to it, plus an additional linker that's missing the drug, the payload. And you'll see that that pattern repeats across the mass spectra, indicating that we can have those linkers appearing in multiple different forms of the ADC. So this is evidence of a process impurity, some spurious chemistry that gave rise to unwanted products. And this would be something you want to monitor as you move through the drug development and stability and batch release process. Okay, let's go to the next slide. This is another instance of data that we can extract using the IDES digest with mass spectrometry. In this instance, we're looking specifically at the FC portion of the antibody. And the IDES digest liberates the 2 FC domains from each other, so we're looking at them individually. And you know, this is where this is the conserved glycosylation region of the antibody exists. And in fact, we can see those glycans attached to this portion of the antibody. In particular, we see 1 glycan per FC subunit. And the annotation here confirms that the same glycans that we saw in the intact analysis is present here with the G0F and the G1F glycans attached to the FC domain. Okay, let's do the next slide. This slide just gives a flavor of what the data might look like if we were to combine the IDES digest with the DTT reduction. In this instance, this treatment releases the subunits completely from each other, so we can look at them all independently. It's the same method here with the reverse phase column, and in this instance, we've set the mass range of the mass spectrometer lower because we're looking at smaller subunits. And here we can get a sense of a better sense of the distribution of the drug molecules and amongst the different subunits. In this data, we see evidence of 0, 1, or 2 drug molecules attached to each light chain. That's on the right, the left-hand portion of the mass spectra there. And then amongst the FD heavy chain fragments, we see evidence of either 0 or 1 drug molecule for each of those subunits. So this is the last data slide that we have for subunit intact data analysis. What we can see here amongst all this data is the overall drug antibody ratio for the ADC, the distribution of those ratios amongst molecules within the sample. And then by using subunit analysis after digestion, we can determine, we can see more features because we reduce the complexity, but also identify where things like glycosylation and drug molecules are attached within the different subunits of the ADC. And this kind of data gives us leverage to monitor these things in a CMC effort to control the quality of the drug. What we can't see here is what happens on a smaller scale, like where exactly in the amino acid sequence is the drug molecule attached. And so on the next slide, I'll just briefly touch on another approach that we use to characterize these molecules, which is a peptide mapping approach. In this strategy, we start with intact ADC, and then we exhaustively trypsinize it to release its component peptides. A nice feature of this approach is that the drug molecules generally stay attached. And so as we load the peptides into the LC and detect the masses by LCMS, we can identify the peptides and look for the masses that correspond with conjugated drug molecules. And that allows us to determine which amino acids in the sample and in the ADC are carrying the conjugated drug. I can show an example of that data on the next slide. Here we're looking at some representative peptide mapping data. I'll start by describing what's happening on the bottom right. You'll see 2 mass spectra there on the bottom right. The top one is a predicted mass spectra. So a software package like Biopharma Finder, it has the amino acid sequence, and it can predict the trypsinization products, and then do an in silico digest and determine what the mass spectra would be that would arise from those products. What I didn't mention on the last slide is that as we use the mass spec to do the peptide mapping, we're doing tandem mass spec here. So we're looking at the intact mass of each peptide, but then allowing the mass spectrometer to fragment the peptide, which allows us to extract mass data that can be used to determine the amino acid sequence of that peptide, and then align it with the unknown amino acid sequence of the antibody. The software will make a theoretical digestion, which is shown in the top mass spectra. And so if the peptide shown in that figure were to be found in the sample, you'd expect it to have the ions, the masses that are shown in red in that top figure. And then as it acquires the mass spectra for the peptides during the analysis, it will fragment them and then monitor the masses that are being detected and see if they align with any of the predicted mass spectra. So the experimental data is shown on the bottom there, and you can see that that the high resolution system gives us the masses that were predicted for that peptide. So we can stay with some degree of confidence that this peptide has been detected at this moment in time, and then aligned in the software to the amino acid sequence of the antibody. If we move back to the left side of the figure here, the mass spectrometer would repeat this iteratively, over hundreds or thousands of peptides in a single sample. And so as it detects the peptides, it maps them to the expected amino acid sequence of the entire protein, and then you can determine a sequence coverage, which is the percentage of the amino acids that are represented let me say it this way, the percentage of the amino acids present in the antibody that are detected in the peptides that are being picked up by the mass spec. So the table on the left shows the coverage in this instance amongst both the light and heavy chains of the antibody. This analysis was able to pick up 100% of the amino acids for both chains. A convenient, very useful feature of this type of analysis also is that if there's a drug and linker conjugated to the peptide, it will produce a characteristic mass shift in the spectra that's detected, and that can be picked up in the data. The linker also might fragment as the peptide is being fragmented for tandem mass spec, and it fragments in a characteristic way, giving rise to a signature ion mass, which is shown in the chemical structure on the bottom left. This drug and linker will fragment where that dotted line is shown, and it gives rise to a signature ion of 547 Daltons. And as you can see in the experimental data on the right, we do see that mass within the peptide mapping data, giving us strong confidence that, that this peptide was conjugated with the drug and linker. Okay, I think that's the last that I have for this portion, and hopefully been able to show how mass spec can be used to really leverage understanding of how the structure and monitor the structure of the ADC. With that, I'll hand it off to Jessica to discuss some more CMC aspects of ADC.
Thank you, Brian, for opening us up with that characterization part of the ADC. I am Jessica Bridges-Weaver. I am a scientific officer at Bioanalytics focused on CMC support. I've been at Bioanalytics now for just over 7 years, but I have a total of roughly 18 years in drug development experience focused on upstream, downstream, and analytical development. Continuing the conversation on control strategy, I want to discuss cytotoxicity bioassay, which is a targeted drug delivery. And we've kind of hit on this a little bit where ADC binds the receptor, endocytosis happens, it intakes the entire complex, proteases cleave the toxin and the linkers, and so there's degradation, and that toxin is then released to the interior part of the cell, and then that released toxin causes cell death. And so to mimic this assay mimic this action, this mechanism of action within an assay, we can perform a cytotoxicity bioassay. This particular case study, it's a cell-based assay where it's very dose dependent on the ADC. So we perform serial dilutions on the ADC. We plate those dilutions. We add target cells. We incubate those over roughly 3 to 4 days, depending on the target cell line. Add a detection reagent, and then we read the detection to measure the biological activity or the intake of the actual ADC and recording cell death. And so in this particular case study, we qualified an assay with 5 different nominal drug concentrations covering a range of 50 to 150%. We did look at linearity and accuracy, and those both passed. As you can see, the more drug that's added to the higher the drug concentration, the more cell death you get, so therefore the lower signal in this particular example. The repeatability passed. Overall CV was less than 15%, and a potency of a force degraded sample was also outside the qualified range of the assay, so it indicates that this assay was also stability indicating. Another type of assay that we also perform for ADCs is an ADCC bioassay to test for effector function. And so in this particular case study, we actually, the mechanism of action for this is the antibody portion of the ADC binds to a target receptor antigen that is expressed on the surface of the target cells or the cancer cells. The FC region is then attracts effector cells, and in this particular example, natural killing cells, and activates that effector cells to release the cytotoxic granules that ultimately weaken the membrane of the cell and kill target cancer cells. And in this particular case, the procedure that we qualified for the potency of an ADC, we have a reference light, so it's a relative potency assay. And so we used target cells, once again, serial dilution of the ADC, We do add an effector cell to this, so it is a dual cell assay. After an incubation period of time, we add a detection of luciferase detection reagent, and we're looking at that in-fat pathway response to indicate that it's been triggered by the effector cells. And this particular assay was validated once again at 5 NDC levels, covering a range of 50 to 200% nominal drug concentration. It passed linearity and accuracy with a slope of 0.98. And repeatability passed with overall CV of less than 13.1, I believe was the exact. So some of the representative data here also shows an indication that the increased drug concentration increases your signal because you have more of the in-fat pathway is triggered, so therefore you have a higher luciferase response. And likewise, if you have less drugs. So some of the representative data there as well. And continuing on our control strategy, looking at impurities of the assays, we also perform HCP ELISAs. And so a standard commercial kit for ELISAs, we actually use an MSD microarray plate. We actually coat that with a capture antibody that's specific for HCPs. We add standards, controls, and samples to that plate. We're adding the detection antibody, which is a biotinylated anti-HCP detection body with a tag. After incubation, that is also measured on the MSD to get the electrochemoluminescence response for that, and then we analyze that data. And just sort of an example, when we look at these assays, we look at a reference standard curve for your HCP reference standard. We'll set limits of detection, quantify an LLOQ as well as an upper level quantification so that we have a working range within the assay. Some of the limitations of an HCP ELISA is lack of identification of individual HCPs. There's potential gaps in detection, expensive reagents. You're also kind of leaned on supply issues, limited antibodies that could be somewhat of a challenge. The USP guidance 1132 has been expanded for an orthogonal method for LCMS, and a mass spec method. And so we're looking at converting and becoming a little bit more identification and quantification specific from our HCP methods and converting those over to a supporting LCMS platform where you do not have to lean on antibodies specific for HCPs. In some cases, if you want to go after a targeted LCMS response, you would need to have stable isotope labeled peptides. But with just general peptide mapping, we can get a little bit better idea of the HCPs that are in your product and monitor them throughout your manufacturing as well as your final drug material. And then I am going to end the CMC conversation. You saw this table earlier as Brian started speaking, and we highlighted a few assays that we were going to cover today. I've repeated that it was with an assay, the portfolio of assays that Bioanalytics can support within CMC to help characterize your ADC. And we look forward to any conversation or any partnership with that.